Hello everyone, welcome back to the class of biotechnology. My name is Dr. Farah Maarfi and today I'm going to discuss with you about the principles of electrophoresis. So first of all, we should know what is actually the electrophoresis is. But before moving forward, if you are new here, please like and subscribe to this channel so that you cannot miss any future video. Now let us start with the general principle of electrophoresis. So first of all, we should know what is electrophoresis. So electrophoresis describes the migration of charged particle under the influence of electric field. Let us understand what does this mean. Suppose we have charged particles, positive or negative ions. These are the charged particles and we want to migrate this particle to a particular distance. Suppose we want these positive particles to move here. All right. So if we put some negative ions here, and we know that the positive is always attracted by the negative ions or negative charge particles. But there is a much distance between these two particles. So to travel, so that these particles can travel with a particular speed to this space or this position, we need to apply some electric field to it. Otherwise, these particles will move in any direction because there is no influence of this negative charge particle to these positive charge particle, because these negative particles are kept very much far away from this place and we want to move this particular charge particle to this direction. So for that, so that it can travel in a particular direction and in a particular position at a particular position. So for that, we need to apply the electric field. And if we apply the electric field, because of this electric field give a speed to these particles and these particles will move in a straight direction towards the negative charge particle. All right. So electrophoresis basically is a technique. This provide a platform to migrate the charged particles under the influence of electric field. All right. Similarly, in biology, there are many biological molecules such as amino acids, peptides, proteins, nucleotides, and the nucleic acids. And these biological molecules possess some ionizable group upon it. And therefore, at any given pH, suppose this is a beaker filled with some solution and this solution is having particular pH. So therefore, at any given pH, the biological molecules that are present here in this solution, they are exist as an electrically charged species. So these molecules in this solution exist as a electrically charged molecules or electrically charged species. Either they are having a positive charge, that is the cations, or they are having the negative charge, that is the anion. So under the influence of the electric field, when these molecules migrate, they migrate either to the cathode or to the anode depending upon the nature of their net charge. Suppose at this position we have all the net positively charged biological molecules. So depending upon the nature of these charge, it should move to this side because of the negative charge. So that it can move to this particular direction, we need to apply the electric field. All right, we need to apply electric field or electric current. So under the influence of the electric field, these charged, positively charged biological molecule will move towards the negatively charged cathode ions. All right. So suppose we have kept a negatively charged cathode here. So under the influence of the electric field, these positively charged biological molecule will migrate towards the negatively charged cathode molecules or cathode ions. All right. But if the charge on the biological molecules are negative, then these negatively charged molecule must move towards the positively charged anode. All right. These negatively charged molecule must move towards the anode. This is the cathode, not to the cathode but towards the anode. So if the charged molecule is negatively, then it must move towards the positively charged anode, and if the charged molecules are positive, then these molecules must move towards the negatively charged cathode. Now the equipment required for electrophoresis consists of two things. One is the electrophoresis unit and another is the power pack. Electrophoresis units are available for running either vertical or horizontal gel system. So this is the image representing the vertical gel system and this is the image representing the horizontal gel system. So by utilizing the vertical gel system, we can run our sample in a vertical direction, and by utilizing the horizontal gel unit, we can run our sample in a horizontal gel system. Vertical gel system is ideal for running the proteins, while the horizontal gel system is ideal for running the smaller samples such as the smaller proteins or the DNA molecules. If we talk about the vertical gel system, then this gel system is having a two glass plates. They are held together in a vertical direction and are separated using a spacer. So these two glass plates are clamped together, but they are held apart by a plastic spacer. So these are the position where the spacers are held. So by the means of the spacer, these two glass plates are held apart so that we have a gap in between these two plates. In these gaps, we will fill our gel. And after pouring the gel in between these two plates, we need to put a comb. The comb looks like this, and these combs provides a well like shape here so that we can fill our sample in these wells after the polymerization of gel. The size of the gel is typically around 8.5 centimeter wide by 5 centimeter high and of thickness 0.5 to 1 mm. Not nm. mm. And when the sample is loaded and all the things are ready, that is the apparatus is assembled and the samples are loaded, then the lower electrophoretic tank, these tanks are filled with buffer, and these buffers surrounds the gel plates, and they also provide some cooling to the gel, because while running the current or while passing the current, there must be the generation of some heat. So to avoid those heat, these buffer provide some cooling effect also. So these are the cathode and anodes by which we can provide the electric field to this electrophoretic system. Now let us discuss about the horizontal gel system. This is the image representing a typical horizontal gel system. This gel system is having a gel cast on a glass or a plastic plate, and below this plastic plate is a cooling plate that allow the insulated surface through which the cooling water is passed to conduct away the heat generated while providing the electric current. And the connection between the gel and the electric buffer is made using a thick wad of wetted filter paper called as wick. So this wick is actually having a pores, or these are capillary in nature, so that they allow the passing of the buffer across it. So in this way, this wick provide a connection between the gel and the buffer. However, these days the agarose gel for the DNA electrophoresis are run submerged totally in the buffer. These days we don't require wick because the gel is completely submerged in the buffer system. Here are the two electrodes that are kept at the end of this electrophoretic unit. The cathode this side, the negatively charged cathode, and the anode this side. The red one is the anode and the black one is the cathode. And at the cathode side, we have our wells so that we can pour our sample into it. Suppose we need to separate our DNA molecule. So the DNA molecule is having a negatively charged upon it, negative charged ions. And when we pour our DNA sample in these wells, so these negatively charged DNA molecule must be attracted by the positively charged anode, and this negatively charged cathode must repel this anode to this direction. And for to move this DNA from this side to this anode side, we need to pass an electric current across it. So for that we need a power pack. So the power pack supplies a direct current between these two electrodes in the electrophoresis unit. All the electrophoresis unit is carried out in an appropriate buffer. In this, everywhere we have a buffer. So these days, the unit which we are using, in this unit we can submerge our buffer completely. We can submerge our gel in the buffer completely. But earlier days, in this typical gel system, we have the compartment of the buffer reservoir, and with the help of the wick we can allow the buffer to travel across the gel so that we can separate our sample using this system. But these days we don't require a wick because our buffer is submerged in our gel is submerged in the buffer. So this appropriate buffer is very important because it is essential to maintain a constant state of ionization of the molecules being separated. As I have earlier told you that pH plays a very important role. So because of this pH, these molecules are having some positive or negative cation or anion forms. So if there is any variation in the pH, then it would alter the overall charged, and hence the mobility of the molecules being separated will be affected. All right. In order to understand how the charged species separates or the moves, it is necessary to look at some simple equation that is related to the electrophoresis. So this is the electrophoretic unit, and these are the two electrodes, and the voltage are being applied to these two electrodes. So when the power supply is applied, or when the power is applied, then the potential difference, that is the voltage, is applied across these two electrodes. So maybe the voltage 1 or the voltage 2. So there are the two voltage applied at the two electrodes. This potential difference, the electrodes, across the electrodes, is generates a potential gradient. What it generates? It generates a potential gradient. This potential gradient is denoted by capital E. So when the potential difference is being applied across the electrodes, it generates a potential gradient, that is E. This potential gradient is the applied voltage, that is the V, capital V, the applied voltage, divided by D. So V is the applied voltage and D is the distance between the electrodes. So D is the distance between the electrodes. All right. So when this potential gradient is applied, the force on the molecules bearing a charge Q will be equal to EQ. Means when this potential gradient E is applied, on the molecules that is present here in the well, so that it can migrate to some particular distance, to particular direction. So these, as you all know, these molecules must be having some charge upon it. So this charge is represented by a small q. So when capital E, that is the potential gradient, is applied on these charged molecule, then the total force according to the newtons on the molecule will be equal to the E into Q. Q is the charge and E is the potential gradient. And it is this force that drives the charged molecules towards the electrode. So due to the presence of this force only, these molecules migrate to particular distance towards the electrode. However, there is also some frictional resistance that retards the movement of these charged molecules. These frictional forces is a measure of the size of the molecules or the shape of the molecule or the pore size of the medium in which the electrophoresis is taking place. And the last one is the viscosity of the buffer. So these are the viscosity of buffer. So these are the four factors that affects the migration of the charged molecules in the electrophoresis unit in between the two electrodes. So these are the factors affecting the migration of molecules. So now if we want to calculate the velocity, that is the small v, of charged molecule in an electric field, we can use the equation v is equal to capital E into Q divided by F. F is the frictional coefficient. Frictional coefficient. So in this way we can calculate the velocity of the charged molecules. But most commonly we use the term electrophoretic mobility. What term we use? We use electrophoretic mobility. And this electrophoretic mobility is denoted by mu, which is generally used for the mobility of ions. So the electrophoretic mobility is small v upon E. So it is the ratio. It is basically the ratio which is the velocity of the ion to the field strength. Therefore, because of this only, even the molecule with a similar charge will begin to separate if they have a different molecular size or shape. Suppose the molecules are of same ion, they are of having positive ions upon it. So these molecules again can be separated based on the different size and shape. The separated samples are then located by staining with an appropriate dye. You can use the bromophenol blue. Bromophenol blue is a dye with which you can stain your DNA sample. Bromophenol blue. Another technique by which you can visualize your DNA sample is using the autoradiography. Autoradiography. But for this, your sample must be pre-radio labeled. Alright. The another most important factor which we are missing is the use of current. Current in this solution between the electrode is conducted mainly by the buffer ion. And according to the Ohm's law, there is a relationship between I, that is the current, voltage V, and resistance R, which states that V by I is equal to R, where V is the voltage, I is the current, and R is the resistance. And according to this equation, it appears that V is directly proportional to I, that is voltage is directly proportional to current. So if we increase the voltage, then the current will increase automatically. So in the electrophoretic system, if we want to increase the migration rate, if we want to separate our sample in a lesser time, so for that we need to increase the voltage. And as soon as we increase this voltage, then according to the applied voltage, the current will be increased. Hence, the distance migrated by the ions will be proportional to both current and time. However, this ignores the major problem, that is the generation of heat. Generation of heat. So if we expose our gel to the increasing voltage or to the increasing current in for a maximum number of time, the porosity of the gel will be affected, and hence it will automatically affect the migration of the charged molecules. So the appropriate current, time, voltage is very important to see or to visualize the separated molecules in a better way. Heating has following effects, like because of heat there is an increased rate of diffusion of the samples, because the pore size of the gels will be affected. The pores of the gel will get bigger because of the heat, or the pores get deformed, and because of that there is an increased rate of diffusion of the samples and a buffer science leading to the broadening of the separated sample. Another thing is that which is affected by heating is the formation of the convection current, which leads to the mixing of the separated sample. Because of the convection current, because of the over current, or because of the high voltage and the current, there is a chance that these samples get mixed with each other. The third thing which is affected by heat is the thermal instability of the sample. Because of the high heat, the samples which are sensitive to the heat may get denatured, like the proteins or the enzymes, they can lose their activity. And the last thing which is affected by the heating is the decrease of the buffer viscosity. Because of the heat, the buffer loses its viscosity, and due to that, there is a decrease or there is a reduction in the resistance of the medium, and because of which in a lesser time the molecules which are being separated travels to a much farther distance, much more distance that they are expected to. So this was all about the general principle of electrophoresis. In the next session we will discuss about the agarose gel electrophoresis and the supporting medium.